For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 3, Episode 6. Highlights from NASH-TAG 2022 looking at combination therapy and sequential treatment and testing issues starts now.
Today on Surfing the NASH Tsunami.
This week, we decided to share some of the transcripts from our NASH-TAG episodes, but with a different point of view. So Wednesday, I shared two specific conversations that might have gotten lost in the back end of episode 2. Today, I'm going to pull together snippets from episodes 2 and 4 to consider a broader issue around combination therapies, thinking not only about a static moment with two drugs at the same time, but also about longitudinal drug and testing strategies. Tomorrow, I'll pull together some discussions about diagnostic testing for drug approval, focusing on digital histopathology, NITs, and some of the economics around them.
Over the past 2 weeks, Surfing the NASH Tsunami compiled over 4 hours of recordings from NASH-TAG 2022, available only in episodes running an hour or longer. This week, Surfing NASH is presenting edits from these recordings, focusing on specific issues or conversations of interest. Listen as panelists from episodes 2 and 4 discuss issues surrounding combination and sequential testing and treatment strategies, today on Surfing the NASH Tsunami.
So the first time this broader issue of combination therapy came up in our podcast last weekend was early Saturday morning as we discussed the first sessions from Friday, where presenters reviewed data from the FXR, FGF19, and FGF21 classes. The first instructive conversation involved the proper role for FXRs and the rationale behind it.
Speaking of the FXRs, doing a deep dive into where we are with that, this is what started the whole NASH movement, at least really getting it on track as far as developing a drug for NASH. Starting off with the Flint trial 6 years ago and then turning that into the Regenerate study. And we all know the issues with that. The therapeutic index has kind of been the challenge with the FXR class, looking at efficacy versus adverse event profiles. In total, there have been about 8 FXRs that have been studied to date. And I showed a slide speaking on the pleiotropic mechanism of action of this drug. And there may be differences within the FXR class as far as efficacy relative to liver fat content reduction, ALT reduction, fibrosis improvement, also some differences in the prevalence of pruritus and LDL increase and HDL decrease. However, as a general rule, I think we can all feel comfortable in saying that pruritus and LDL increase are generally a class effect. Having said that, there are still FXRs in play. There are some that are no longer being developed. I mentioned already that Intercept beta-cholic acid is still working to try to get FDA approval and resubmitting their package of data with additional safety data sets as well as efficacy. So it's still pending in that regard. I don't think the overall FXR class is a non-starter anymore. I do think that the pendulum has shifted from the thinking that the FXR class will be the backbone of therapy for NASH. My comments were, if we can reduce the dose of the FXR, maybe mitigate some of the adverse event profile and add it to another agent, we could boost the efficacy of the second agent and synergistically get a little further down the road as far as improving the histopathology of these patients. Rohit was moderating the session and asked several of us what we thought the biggest challenge with the drug was. Myself and Vlad thought pruritus was the bigger issue here. My reason for saying that was mainly my patients are asymptomatic. And when I put them on a drug and 2 weeks, 4 weeks later, they're coming to me complaining of itching to the point where they're complaining of itching, and I'm not asking about, do you itch? At that point, I can almost assure you that most of those people will eventually quit taking the drug or would not take it with any rigor. Now, that was countered by many other people saying they thought hepatobiliary issues were the biggest concern with this class of drug, gallstone formation, issues with potential DILI. There was a robust discussion, good discussion, I would say, but there was disagreement as to the severity of hepatobiliary impacts on this class. But I would just say that that is one of the challenges that's going to have to be dealt with as we move forward. And quite frankly, that's my whole reasoning for lowering the dose of the FXR and pulling it in combination with something else. At the end of the day, the ideal therapy, and I showed this slide also, is therapy that has histopathologic benefit across the board, but also there's benefits on many of the extrahepatic complications that our patients suffer from liver fat content reduction to lipid lowering to glycemic control and weight loss. So there is an opportunity for FXRs to come in and augment the histopathologic benefits, but just having this hashtag real talk conversation this morning, there's not a lot of benefit on glycemic control, certainly not benefit on lipid lowering or weight loss to any significant degree. So again, I think at the end of the day, they're not dead, but they're best suited for combination therapy in my mind.
So Stephen, you made a really interesting point about this, which is, this balance between efficacy, safety and tolerability. And I think we need to keep safety and tolerability as almost two separate headings rather than one. But it also strikes me with OCA that this is not OCA, all FXR agonists. This is a class where actually we are needing personalized medicine approaches, because what we're currently doing is assuming that you give an FXR agonist, you have the same response in all individuals, and that's clearly not the case.
So can I just keep us reminded that the FXRs, I just don't want to, you're right, it's good for combo, but I think the field is going to all be combo, like type 2 diabetes. And with a drug like this, apparently, like if you go higher in the dose, you get side effects, no matter what generation you're in. Maybe the second generation is better, less itching, but I think that it's dose related very much, and we talked about this yesterday. We also need to keep in mind the other agents' side effects, like the FGFs, the diarrheas, the injectable drugs, and I think a lot of drugs needs to be combined with agent like FXR. So to me, is the FXR, I will still say it's probably a main player, and everything is going to be combined at the end of the day. It's just like, what's the right combo? And I really like Quentin's point about personalized medicine. Maybe we'll like genotype people at the end and start therapy based on that.
This is one of the directions as a whole. We all agree that combination therapy is likely to be the direction of travel here. It's also then about personalizing that choice of combination to the individuals. This is something that actually is going to be increasingly important for us to start handling and building into our phase 2 and phase 3 studies so that we really understand the cohorts.
After a comment from Louise Campbell about the value of starting to assess precision medicine in the context of who will execute and then benefit from diet and exercise, Mazen Nuruddin went back to Quentin's earlier point.
I will go back to the precision medicine that Quentin talked about. We have done that before with other diseases, hepatitis C, or genotype 1 through 6. So I won't be surprised one day we'll say PNLP panel is that, TMS6F is that, start such a therapy. We need to go there further and start looking at response. I don't recall any more than a study or two that looks at responses with genotype, and no one looked at the placebo with genotyping. So I think this is a wide open area that needs to be explored.
So you're absolutely right. One of the few studies that springs to mind on it actually was one done with elafibranor, which was presented at EASL by ourselves. And actually what you saw there was it was non-significant, but it was woefully underpowered, but a trend towards greater efficacy in PNPLA3 carriers, which, if you think about it, is entirely consistent with the fact that we know that carriers of PNPLA3 are more likely to have a greater beneficial effect from diet and weight loss and lifestyle change and so on. So it's this idea that on one side, carrying the genetic variant makes you at greater risk of the disease, but also you've got more to gain by an intervention.
And maybe it's not just the PNLP3, maybe a genetic risk score needs to be thrown in this because it was not significant. Maybe the other genes are also playing a role, and you need to, you need to come out.
We're at the beginning of that road, right? I mean, that is the point.
And
we're in the early stages of that one.
I would say is that sponsors really need to be building this in right from the get-go in the studies. There's often a concern that maybe one's going to unearth something that one didn't want to know, and I think that's a unfortunate way of thinking about it. I think it's much better to build this in, have knowledge, accept that there will always be tolerability issues, downsides to any compound, but actually look to see how you can stratify and mitigate it.
Indeed, you can argue that you can risk stratify randomization in the future based on those, rather than one right now, the type 2 diabetes. I mean, it's an important factor, but but in the future, you might have more success if you genotype well and randomize per genotype.
So far, the group had only been talking about FXRs as a class and maybe some theoretical issues. But when Stephen shifts the conversation towards FGF19s and later 21s, the entire concept of combination therapy starts to gain substance and multiple dimensions.
I think we all agree that this compound has biological activity in the liver. And just following on the tails of FXRs, and I thought this was kind of cool that it happened right after the FXR talk, is, FGF19 works through FGF beta-klotho 1C and 4C, or 1C and 4. And so there's this idea that FGF19 agonism is going to work on peripheral adipose tissue insulin sensitivity and downregulate free fatty acid flux to the liver. And maybe that's the reason why you see rapid liver fat content reduction in PDFF. Then there's the whole other action through FGFR4 and its effects on bile acids. And is that the bigger play here? What's the role of the bile acid inhibition relative to the role of decreasing free fatty acid flux? If we can learn more from that, we can take the lessons learned from the FGF19 class and apply them to either combination therapies that we have in play today, or as we begin to design novel therapies in the future to get around some of these issues that we face with LDL increase or whatnot.
By the time we shift fully to FGF21s, two entirely different dimensions around combination therapy come into play. Stephen starts by pointing out the incredible range of diversity of agents within the class, highlighting the point that we're just at the beginning of figuring out what we need to learn about FGF21s. Still, he expresses real excitement about their potential and is puzzled why colleagues might not feel that way.
But I think with the 21s, we are literally at the beginning of understanding this class fully, and it is the wild, wild west. Mm-hmm. But I'm super excited about it. I guess I walked away a little bit yesterday feeling that there wasn't that robust excitement with the FGF21s. And maybe I took away a different feeling than others, but I am excited about the 21s, despite the fact that it's injectable, despite that the fact there are some tolerability issues with GI. I think this class has an opportunity, when used in the right patient population, the more advanced patient population, as an induction therapy to get these people under rapid control. We are going to see extrahepatic effects that are very, very promising for some of these drugs, whether it's weight loss, lipid reduction, glycemic control. We've seen that with this class. Mm-hmm. So I'm high on the 21s coming out of NASH-TAG 2022.
I totally agree with that. I mean, my point was between the FXR and FGFs, I think they're intimately close to each other. I think that axis is very important, and we'll be talking about it. Then TH-beta is something different. And you have, for instance, GLP-1s is daily week injection. It's great. You lose weight, but to Stephen's point, the FGFs has direct effect on the liver, and that's important. And that axis is in the pathophysiology of the disease, and we need to address that axis and that underlying pathophysiology.
So I totally agree. One of the things I heard from 3 or 4 people yesterday, because I just interviewed people because I don't know enough to be dangerous anyway, but I know enough to ask questions. 3 or 4 people raised the concern that peg-belfermin falling apart in 48 weeks scared the stuffing out of, and adiponectin. And those were the 2 things I heard that people took out of yesterday, maybe more than they went into it with. Now, to have the commercial and medical sophistication that you just exhibited to say, it's maybe an induction drug, it may only go 6 months, right? And then we might run out of room with it. But for a lot of patients, those 6 months are really going to matter. That, I think, is what everybody needs to start thinking about. Along with combination therapy isn't only which drugs are you taking at the same time, but if you plot a flight path for a patient, right, as we go from Wright brothers to 5th generation strike fighters,
right?
As we plot a flight path is also temporal. It's got a time dimension in it.
Yeah, so this is where I will get kind of cross-eyed with some of my colleagues, because there's this thought that if a drug is not moving fibrosis, it's because we're not treating long enough. And that's a comment I hear over and over again. But I really believe that fibrosis is more dynamic than what people give it credit for. And until you become portal hypertensive cirrhotic, we can move the needle on fibrosis quicker with the right drug, hitting the right, and it may be personalized medicine. It may be it takes the right drug in the right patient to move the amount of fibrosis as quickly as we'd like to move it. But I think the 21 class, look, we may have issues with ADAs, with anti-drug antibodies. We may have issues with tolerability. But if we dose it in the right patient for the right length of time, I think we could have the effect that we want, not just on the liver, but on the extrahepatic benefits, and transition them over to something else for the long haul.
So by the time we get to the end of that conversation, you can see two dimensions emerging. Question number one, would we want to give two different drugs at the same time to achieve two different purposes with lower doses, the kind of thing we talked about with FXRs, maybe FGF19? And then number two, would we like to start out with one drug that has tremendous short-term power, but then over time shift to something that might be more sustainable? Now, when the conversation shifts from Stephen's lead, which was about drugs, to Quentin's, not surprisingly, topic shifts to diagnostics. And even within the diagnostic topic, though, one focus becomes the longitudinal use of diagnostic testing to achieve different diagnostic goals efficiently throughout the process. As you listen, hear how standard this thinking is for testing, as compared to long-term medication strategies or pathways that rely on, as a single course, doing different things at different times.
So for our listeners, from your perspective, looking at these huge data sets, ELF and FIB4, are you, I'll just throw an action item out there, are you thinking today that you would initially screen with FIB4 if you have an indeterminate or positive value to FIB4, that you would chase that with an ELF because you like it more as a second-tier test to stratify the advanced disease, as opposed to an initial test where you're really trying to maybe get at excluding patients?
Absolutely. So I think this is a really important point. FIB4, it's very cheap, it's very easy to apply. It is therefore very good to rule out disease, and it allows you to enrich the population that goes forward and improve the performance of a second-line test. Now, that could be ELF, it could be FibroScan. You can argue about what you use next till the cows come home, but actually it's how you game the situation to improve the performance of the second-line test.
And
It's not hard. It's not hard anymore.
so also,
Bard has got a place in our hearts, Stephen.
also for FIB4, I totally see it this way. And I also want to keep us reminded that correlation with outcomes is very important. And ELF is doing very well with that, VCT and MRE. I want to remind people of the excellent JHEP paper that came exactly. So the outcomes with FIB4, almost half of the patients that were in the low or indeterminate zone had events, if I remember correctly.
I think the thing we've got to take away is that any biomarker that accurately stages fibrosis will have prognostic value, because the presence of fibrosis has prognostic value. So, it's true of FIB4, it's true of NAFL fibrosis score, it's true of ELF, it's true of FibroScan. It will probably be true of any other good fibrosis biomarker that comes along.
So that pretty much wraps up our conversation about combination therapies and longitudinal use of drugs and diagnostics from episode 2. In episode 4, our wrap-up with Amy Articolo, Erin Quirk, Ian Rowe, and Rachel Zayas, the same point comes up again. First from Ian, in terms of multiple goals during therapy with antifibrotic drugs or fibrotically balanced therapy. And then from Amy, in terms of, from a corporate perspective, how do you think about combination therapy as a way to solve problems? Have a listen.
The point that he made about unmet needs in fibrosis was, when we think about antifibrotics, we're thinking about drugs that are going to block the progression of disease. And what he was really talking about was trying to understand resolution of fibrosis more clearly so that you could target the other side of the balance. So that once you've switched off injury, or maybe even without switching off injury, then you can promote the resolution of the scar, allow the fibrosis to regress, and for the patient to have a better outcome. I guess putting those two things together, the team in Edinburgh are running studies looking at macrophage-based cell therapies to try and promote scar resolution, really as proof of concept that can be done. There's lots to be seen through the approaches complementarily, looking at both progression and regression.
Yeah, and I think that your point is incredibly well taken. In our drug development strategies, we have tended to say we'll knock out the upstream drivers of stellate cell activation, and vicariously we'll wait for the liver to begin to heal, rather than targeting therapies that could actually promote wound healing in addition.
And to that end, I'd love to add, I really appreciate those talks, because I'm a big fan of, let's get to the core of the problem, right? What are we really looking to drive and really solve for? And so that for me, I felt like it was the aha moment of the talk that my Michael Charlton gave regarding combination trials and his hearts along, what he would like to see, because it resonated quite a bit with me, not only as an individual, but also, from a company that's striving to look at combinations, but also to hear Scott talk about that and the upstream effect. And so it makes you pause and go, okay, so what are we really trying to do? What is the greatest impact for the greatest good in a very patient-centric approach, thinking, what is the patient going to be willing to take to improve how they function, how they feel? And what can we as collectively do to really get us from point A to point B so that we're able to put that therapeutic and that potential combination therapeutic in the patient's hands?
This concludes episode 6. Tomorrow, we'll be back with episode 7, where we will take a look at issues surrounding NITs and the fireside chat and the path towards making it easier to go beyond the biopsy. I hope you enjoyed this episode. I hope you come back for episode 7 tomorrow, and look forward to seeing you soon. Stay safe, surf on. See you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.